Welcome, everyone. My name is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. I wanted to welcome you to this first in a series of what we're calling our award winners series, and we're very excited to start this off and to partner with the European Society for Toxicology in vitro. First, I'm going to do a bit of brief housekeeping related to the webinar. As you might have heard, the webinar is recorded, and ASCCT and NSTIV members can have access to the webinar recording, as well as all of our past webinar recordings. Each presentation is going to run for about 20 minutes, and we'll have 5 minutes for questions. So if you have a question for the presenter, please use the question module to type in your question, and I will read any questions to the presenter when they're finished. We will also make a PDF of the slides of the presentations available to attendees a few days after the webinar. And I also wanted to mention that we have 2 webinars coming up in the next couple of months. Additional early career scientists who have won SDIV or ASCCT awards last year. So I hope you'll register for both of those. You can go to ascctalks.org/webinars to register for those. I'm going to introduce our presenters, and then I'm going to invite SDIV President Matthew Vinken to talk a little bit about their award. So first, we're going to hear a presentation by Maria João Bessa, and she holds a Master in Bioengineering and is currently a PhD student at the Portuguese National Institute of Health and Institute of Public Health from the University of Porto. Maria João's research interests are in the field of in vitro studies in nanotoxicology, and her present work is based on the toxicity impact of nanoparticles derived from the ceramic industry. Our other presenter, will go second, is Tom Vollicht. Tom first got into contact with in vitro toxicology during his PhD work at the Helmholtz Center for Infection Research, setting up a new liver cell system for toxicology assays and infection research. He now works for InScreenX, developing expandable cell systems from primary material for toxicity and bioassays as an alternative to animal models and primary cell material. So Matthew Vinken, you should be unmuted, so please go ahead and talk a little bit about the awards.
Okay, thank you so much, Christy. So depending on the time zone you're in right now, good morning or good afternoon to you. It's very nice to see, well, not just to see you, but at least to know that you attend to these webinars. So as Christy rightly mentioned already, this is the first of these kind of webinars, in the sense that you might know that SDIV organizes a number of events every year, and we do have the conference every 2 years. So the previous conference, the most recent ones, took place in Berlin, in Germany, and that was in October 2018. And during that conference, we have granted a number of awards, especially to young investigators, for either their oral presentation or poster presentation. And we used a number of criteria, among which excellence, also the relevance to the field of in vitro or in silico toxicology. So then the idea came, because you might know we also have a memorandum of understanding between the American Society, so the ASCCT and SDIV. And as part of this memorandum of understanding, we decided to set up a number of joint initiatives, among which does these webinars. So I'm very honored, actually, that we now can give the floor to these 2 excellent researchers, which already have been introduced by Christy. And again, as already mentioned, also we do hope to welcome you during the next webinars, which will take place in February and March. So that was it from my side. I think we can give the floor now to our 2 young investigators.
Great, thank you, Matthew. Now, without further ado, I will go ahead and introduce our first presenter. Give me a brief moment to switch over here. So we're first going to hear from Maria João Bessa.
Hello. I hope that everyone can hear me.
Yep.
I'm going to start
the slides.
I'm going to start my presentation on pairwise toxicity evaluation of ceramic nanoparticles exposure in human alveolar epithelial cells at submerged and air liquid cultures. This work is under the framework of my PhD, and will also contribute for the ZrO2 project, the Aeronet ZrO2 project. Starting with a brief introduction, the ceramic industry is a growing industrial sector that has been offering a wide range of applications with a great impact in our daily lives, since traditional materials, for example, bricks and tiles, that are used in houses and buildings, to more advanced materials such as electronic and magnetic components. These are some illustrative images of the ceramic industry that are from the ZrO2 project. The majority of ceramic companies, as well as the most qualified employees, are located in Europe. In fact, around 200,000 people work in the ceramic industry in all countries of the European Union. In addition, 23% of the ceramic worldwide production is from the European Union ceramic manufacturers, reaching a production value around 28 billion euros per year. So with this data, you can understand how the ceramic industry plays such an important role in the European economy, as well as in the global economy. Over the last years, nanotechnology has been offering a great potential to develop new functionalities and innovations in the ceramic industry processes and products. However, nanoparticles may be released in the workplace environment, meaning that both materials and technologies can be sources of nanoparticles exposure to the workers in ceramic occupational settings. And it has been well stated in the literature, the respiratory system is one of the major routes of interest of nanoparticles, and in occupational settings, inhalation is a predominant route. And the smaller the particles, the easier they can penetrate in our body, specifically deeply into our lungs. However, their real impact on human health is still poorly understood. Over the years, studies on pulmonary in vitro cell toxicity of nanoparticles are usually performed under submerged culture conditions. However, in vivo, and specifically in humans, the exposure to airborne nanoparticles occurs at air-liquid interface conditions and not in immersed conditions. So, the air-liquid interface exposure is a more realistic exposure scenario for nanoparticles inhaled into the lung. Taking all into account, the main goal of the present work was to investigate the in vitro toxicity of three derived ceramic nanoparticles, zircon oxide, zirconium oxide, and antimony tin oxide nanoparticles, in a human alveolar epithelial cell line that is used as a primary target during inhalation exposure. For the present work, two in vitro toxicity approaches were performed. Cell lines in submerged cell cultures and polarized cell cultures at air-liquid interface conditions. The chosen cell line was the A549 cells that are a human alveolar epithelial cell line. Starting with the submerged conditions, this human alveolar epithelial cell line were adhered to a flat and rigid substrate where they allowed to adhere. And during 24 hours, these cells were exposed to the selected ceramic nanoparticles at a range concentration between 5 and 150 micrograms per square centimeter. In parallel, we also performed a set of negative and positive controls to validate our data. At air-liquid interface conditions, cells were seated at a permeable membrane in the transwell insert, where they are allowed to adhere as a monolayer of polarized cells. During exposure, the apical medium was removed and cells were exposed to aerosolized nanoceramics using the vitro cell automated exposure station for 2 and 4 hours. Each time point refers to a different concentration. In parallel, we also tested a set of negative controls, one being the incubator control, where cells were presented in the incubator, and the others where cells were exposed to clean air that we denominated as exposure control. This is a representative image of the vitro cell system used in the Netherlands National Institute for Public Health and the Environment. For the toxicity evaluation, relevant biomarkers were assessed regarding cyto and genotoxicity. In terms of cytotoxicity, 2 assays were performed. The first one was the LDH release to assess the plasma membrane integrity of cells, while the WFC1 metabolization assay was performed to assess the cell viability. In terms of genotoxicity, it was performed the COMET assay, where the alkaline version allows us to evaluate the primary DNA damage, and the enzyme-modified COMET assay using the FPG enzyme was used to assess the oxidative DNA damage. Moving on to our results, and starting with the results on submerged cell cultures. In terms of cytotoxicity, no major deleterious effects were observed after 24 hours exposure to all selected nanoparticles for this study. Nonetheless, in parallel, we also performed some interferences studies, since at the literature it has been stated that nanoparticles can interfere with the assays. And in our case, you can see a marked interference of the seroxide nanoparticles with the LDH assay. You can see that for the concentration-dependent increase of LDH release, which can be due to the position of nanoparticles in the bottom of the well, preventing the release of the LDH to the supernatant. In addition, we also expose cells to the highest concentration of seroxide nanoparticles plus a positive control with Triton, and we observe lower levels of LDH release, which can be due to not only to optical interferences, but also catalytic interferences of these nanoparticles with the assay. Regarding cell viability, no interferences were found for any of the tested nanoparticles. In terms of genotoxicity, 3 non-cytotoxic concentrations were tested. And after 24 hours of exposure, for the highest concentration of all tested nanoparticles, you can see a significant increase of the DNA damage of cells. However, in terms of oxidative DNA damage, no major effects were found, with the exception of the cells exposed to the highest concentration of antimony tinoxide nanoparticles. We could also see a significant increase in the oxidative DNA damage of cells. Moving on to the results on the polarized cell cultures, and starting with the cytotoxicity, we assessed 3 endpoints: before and after exposure, and 24 hours after the exposure. And regarding the seroxide nanoparticles, as expected before exposure, no effects were found in terms of LDH release. However, after 4 hours of exposure, we could see a significant increase of the LDH release in comparison to the exposure control, meaning that the plasma membrane integrity of plasma membrane of cells lost their integrity. In parallel, we also performed some interference studies, and in contrast of what we observed at submerged conditions, no interferences were found with the LDH assay. After 24 hours after to the exposure to the aerosolized nanoceramics, we could see significant increase of LDH release in the exposure treatments and exposure control with the incubation in comparison with the incubation control, and we could see a significant decrease in the same treatments in the cell viability. Regarding zirconium oxide nanoparticles, similar effects were observed, meaning that before exposure no effects were found. However, after 4 hours of exposure, we could observe a significant increase on the LDH release in comparison with exposure control. In parallel, we performed the interference studies, and once again, no interferences were found. 24 hours after exposure to the aerosolized nanoparticles, we could also see that the plasma membrane integrity and the cell viability of cells were still damaged, still affected for both exposure control and exposure treatment, meaning that the flow rate with or without nanoparticles could affect the cells even 24 hours after the exposure. Regarding the antimony tin oxide nanoparticles, once again, before exposure, no effects were found, and significant effects were not found also after exposure. However, we could see an increase of the LDH release 24 hours after exposure to these antimony tin oxide nanoparticles. Regarding the interference studies, no effects once again were found, and 24 hours after exposure, we could see that cells could see an increase in the LDH release and a decrease in cell viability of cells that were previously exposed to the aerosolized nanoceramics in comparison with our incubator control. Regarding our genotoxicity results, it was assessed 24 hours after the exposure in the polarized cell cultures at their liquid interface. And after 4 hours of exposure to the aerosolized ZrO2 oxide and zirconium oxide nanoparticles, you could see a significant increase in the primary DNA damage of cells in comparison with exposure control. Whereas we could also see a significant increase in the primary DNA damage after exposure to the zirconium oxide nanoparticles, regardless of the time of exposure, in comparison with incubation control. In contrast, no effects were found regarding the oxidative DNA damage after exposure to these aerosolized nanoparticles using the vitro cell system. As conclusions, you could see that different toxicity effects were observed in both types of culture conditions after exposure to the nanoceramics. However, when cells were prone to more pronounced effects after exposure to the aerosolized nanoparticles at air-liquid interface conditions, specifically a decrease in the plasma membrane integrity and in cell viability, and also an increase in the primary DNA damage after exposure to the aerosolized ZrO2 oxide and zirconium oxide nanoparticles. In addition, we could also observe that these conditions with their liquid interface, using their liquid interface, were less prone to interfere. Aerosolized nanoparticles were less prone to interfere with our cytotoxicity assays, in contrast with the submerged conditions, where we could see a clear interference of the ZrO2 oxide nanoparticles with the LDH assay. The observed differences could be also arise from the different deposited doses different conditions, submerged or air liquid interface. Nonetheless, our results can highlight the potential effects, potential health risks associated with exposure to these derived ceramic nanoparticles, especially when are in the form in aerosols. To conclude, I would like to acknowledge my host institutions, the National Institute of Health and the Institute of Public Health from the University of Porto. I would also like to acknowledge the SAFE project from which these results will contribute for, which I believe will be of great importance. And also, I'd like to acknowledge the Institute for Public Health and the Environment for the set of experiments performed in terms of their liquid interface conditions. I would also like to acknowledge the Portuguese Foundation for Science and Technology for my PhD scholarship. More importantly, and to conclude, I'd like to acknowledge the European Society of Technology in Vitro and also the American Society for Cellular and Computational Technology to give me the opportunity to present in this webinar. In addition, I'd like to acknowledge the Applied In Vitro Toxicology for the award in the 20th International Congress on In Vitro Toxicology in Berlin in the STIV Congress. Thank you very much for your attention, and if you want to ask any questions, I hope I can answer.
Great, thank you very much, Maria João, for the presentation of that work. Just as a reminder to everyone, if you have a question, please type it into the question box and we can get it answered. Perhaps in the meantime, I have a question for you. What are you working on now in terms of follow-up for the project?
And now you move on to 3D cultures using epithelial model, mucillar, and we tested not only engineered nanoparticles, just as the one that I showed you here, but also process-generated nanoparticles derived from the ceramic industry. And so far, the results are being really interesting.
Great. We don't have any more questions, so I think we'll move to the next presentation. Oh, sorry.
Okay.
One just again.
Okay.
The question is, did you consider whether or not the enzymatic activity of LDH was affected?
Yes, this is one of the possible interferences of the nanoparticles with the assay. However, we only tested possible optical and catalytic interferences, but for sure that are very important to perform the enzymatic ones. We have performed that already in our lab in previous works, and we also see other type of interference with using other nanoparticles. However, for this work, these were the only interferences studies that we prefer so far. But in the future, we hope to perform also the catalytic interference to make sure what are interfering with the assay.
Okay. And is there a method to normalize the cell exposure between the two systems? So looking to compare.
Yes, if you can. Can you repeat? Sorry, I couldn't understand.
Is there a method to normalize the cell exposure between the two systems?
So far, I didn't perform, but in the future is one of the things that we also want to perform.
Okay. And another question, does the DNA damage observed correlate with increased incidence of cancer in ceramics industry workers?
So far, it has been reported specifically some pulmonary diseases, not specifically cancer, but some bronchitis and other type of diseases. However, in the long-term exposure, the workers can present some type of I'm guessing lung cancer that can be due to this exposure to nanoparticles or fine particles derived from ceramic industry. And these results show that in reality, you could see, in fact, an increase of DNA damage. So it's a possible effect, for sure. I don't know if I answered the question.
Yes, I would think cancer would be sort of the longest term apical endpoint.
Yes.
And that we might try to look for more events. Yeah.
As far as I've seen, there are reports, but they're describing more short-term exposures. Mm-hmm. And so far, it cannot affirm that there are cancers that are directly related to the ceramic workers. But this project, I think, will help us to understand that and create some mitigation strategies to overcome these possibly risk outcomes.
Great. Okay, well, thank you again, Maria João, and
thank you very much, Christy.
I'm going to, she'll stay on the line in case there was something that someone didn't get a chance to ask. We can switch back. But we're going to go now, if you'll permit me to change presenter.
Yes, of course.
Tom Vollert, Dr. Vollert.
Okay, well, thanks very much, Christy. Thanks very much, Maria João. Yeah, so welcome everyone. As Mathieu said, good morning, good afternoon, depending on where you're listening from. First of all, I'd like to thank the ASCCT and also the SDIV, especially Mathieu and Christy on behalf of these societies for like setting up these webinar series. I think they're like an awesome possibility for junior and young scientists to kind of get their science out and talk about the science. Also, thanks very much for the to the ZrO2 Society who kind of sponsored the award, which is the reason I have the honor to present here today. Yeah, so I'm going to talk about how we can use immortalized or expandable cells as a source material for reproducible in vitro toxicology testing based on cell systems. And the stuff I'm going to show is a bit of a mix-up between my PhD project and the work I've done here at the company in Screenix now, and also some colleagues have done at the company as well. And I'm happy to answer any questions, so yeah, just leave them in the chat section. Okay, so to start with, I'd like to start with why do we actually need cells? And I think Maria João did a perfect kind of introduction to why we need cells. So they are like really urgent questions. For example, the human organs on the CHIPS approaches, they need a cellular source. And as I said, Maria João just talked about how we use lung cells to evaluate the toxicity of, for example, nanomaterials that are really everywhere right now. But also think about how drugs induce liver injury. So we need to test this at some point. Or if you think about the huge endocrine disruptor screening programs that have been set up recently. So they all have one thing in common, and that is they need some sort of cell source to do their in vitro assays and to do high throughput screening, especially if you look at the 3R aspect of reducing, refining, and replacing animal experiments. We need some sort of reliable cell source to do all the tox assays that we want to do. And just to kind of briefly talk about what kind of cell sources there are and why we think we need an alternative cell source. So you can just really simply put all the cell sources in a very simple two-dimensional graph where you have the ease of use and the biomass you can produce, and you just need them to screen large compound libraries. For example, if you look at the EPA endocrine disruptor program, they want to screen 10,000 compounds. So the cells need to be high throughput compatible, so you need just an easy to use and a very readily available cell system. And of course, the cell source needs to be kind of relevant. So you want the assay or the cells in your assay to have huge predictive value, so you can translate the findings really easily from in vitro to the in vivo situation. And just looking at a few cell sources, so obvious cell source number one for a lot of assays are cancer cell lines, and Maria João just talked about the A549 cell line that is widely used as an alveolar model. And of course, they're really easy to use. You can produce a lot of biomass, but usually they're not really that relevant. On the other hand, you have primary cells, which are much more relevant in cancer cell lines in terms of the predictive value, but sometimes the access to these cells is really limited, and the biomass you can produce from these cells is really limited as well. Then you have cells that are derived from iPS, so induced pluripotent stem cells, which sit kind of in between. So they're relatively easy to use and produce, and they're relevant enough, certainly more relevant than most of the cancer cell lines, but still the kind of differentiation protocols are really expensive and cumbersome for some cell types. As an alternative, what we think would be a really cool cell source would be relevant cell lines. So they combine the ease of use and the biomass you get with cancer cell lines, but you also combine them with the functionality and relevance of primary cells, and we hope to get these cells to a level where they're more relevant than the cells you derive from iPS cells. Okay, so how do we do this? How does it work, and what do we do different compared to other immortalization kind of approaches? So how it works is, so we get some sort of tissue, just for as in few examples I've listed here, like liver tissue and intestinal tissue. You would isolate fresh primary cells, and to these primary cells we would add an lentiviral library that contains 33 genes. And that's the trick. So we don't really have one gene or one kind of growth and expansion pathway that we tackle. No, we have 33 genes that kind of manipulate the cell cycle, that control apoptosis, that confer stemness, and all have something to do with growth. So we have 33 genes that all have something to do with the growth of cells and expansion potential. So what we have is this lentiviral library, and we add these lentiviruses to the freshly isolated primary cells. Now what we then get is some of the cells will receive a gene combination that leads to proliferating cells. Some of them will receive a gene combination of genes that just lead to no proliferation, or some cells won't receive any of these expansion genes and they will just go into senescence, so into cellular crisis. And what we then do is we don't really focus on the cells that don't grow. No, we are only interested in the cells that do grow. So, and these cells necessarily need to have received an advantageous gene combination, in the sense of this gene combination allows them to grow. And just as an example, here are a few cell lines that we generated. So in red, you always see the proliferative potential of controlled primary cells, and in black, you see some cell lines that we generated. And for example, if we look at chondrocytes, what you can see is you have the control cell lines. They do proliferate, they do grow, but they grow way too slow to do any high throughput screening. If you look at the expandable cell lines that we generated from these primary cells, you can see, and on the y-axis, you have cumulative population doublings. So you can see we have a good proliferative potential, and this is true for if you culture the cells for 100 days. And you have some cells, for example, hepatocytes, where the primary cells stop to grow really fast after a couple of days. They just don't proliferate and just sit there. And if we generate our expandable cells from them, you can see like a good growth potential. And just to give you an idea of like the universal approach that we have with this huge library. So we've immortalized cells from many different tissue sources, including lung epithelial cells, intestinal cells, chondrocytes, also some neuronal cells, so astrocytes and pericytes, and as I mentioned already, hepatocytes. And the good thing about these growth and expansion pathways is they're really conserved among species. So the genes that we have, although they are murine origin, human origin, and some viral genes in there as well, they work for different species. And I think right now we're at 9 or 10 species where we successfully immortalize cells from. And it's really exciting because then you can also have the comparison between certain species that are now used as animal models and then the human cells to see hopefully that human cells are much more relevant. Yeah, so that was the first thing. So what we get is proliferating cells. That's what I showed you. So these cells proliferate and are expandable. But now the really important thing is, especially if you think about in vitro toxicology, you want to have functional and relevant cells. So what we do is we then look at markers, barrier formation for epithelial cells, enzymatic activity. And of course, they in the end need to be suitable for in vitro toxicology. And I've picked 2 examples to kind of illustrate the in vitro toxicology potential of the cell systems. These are hepatocytes on the one hand and alveolar epithelial cells on the other hand, which might be an alternative to the A549 cells that have been around for a few decades right now. So looking at the expandable hepatocytes that we generated, just looking at some basic functions. So I told you, so these cells and these cell systems that we generate, you can see in green here, they do proliferate, whereas the primary cells that just sit there don't proliferate, and then after some time they just go into senescence. And the cell line that we picked here, you can see it only carries 2 of the expansion genes. So although we added the library of 33 genes, apparently these cells only need 2 of these genes out of this library to grow and perform really well as a hepatic cell line. They do have some basic hepatic functions, so they do express albumin on the mRNA level, on the protein level, they do store glycogen. And what's really exciting is that if you look at these bar here at the grayish points, this is normal 2D standard culture. And you can increase the expression of albumin if you culture them in a 3D environment, which means these cells are still responsive to kind of a differentiation stimulus. Now, looking at the really important thing here, and this would be phase 1 metabolism. So these are the famous CYP enzymes, which are responsible for the conversion of toxic substances into more readily soluble substances. And this is what sometimes leads to drug-induced liver injuries, if you have substances that are converted by these enzymes into toxic metabolites. So these CYP enzymes, they need to be there and active to have that as like a really good cell system to use. And we looked at 2 of the most important of these enzymes that do metabolize quite a lot of the toxic substances. And what you can see is there is mRNA expression. And interestingly, if you look at the whitish points against the green points, you can induce the expression if you add a toxic insult, in this case 3-methylcholindrane, or for the other enzyme rifampicin, you can see you can induce the expression, and that's how it should be, because then this toxic sensing is still active. More importantly, they also show not only expression, but also enzymatic activity. So these enzymes, we just showed this with the luciferase-based assay, they are enzymatically active. And again, if you add the inducer, you can see an increased activity where you couldn't detect it before, or for the other enzyme, you could induce the enzymatic activity if you add this toxic insult. So the sensing system of these toxic substances works in these cells. And to illustrate this is a viable system to test toxic substances, we added multiple concentrations of APAP, better known by its brand name paracetamol, to these cells. And paracetamol per se is not really toxic, but in higher concentrations, the clearing system, so the CYP enzymes are overloaded, and there's a production of a toxic metabolite, and this is what leads to liver damage. That's why it's really toxic to take high doses of paracetamol. What you can see is that we can replicate this behavior. So after a certain concentration, we see the cells die. So this is a standard viability assay because of these toxic metabolites that they themselves produce. And we could see that the enzymatic activity that is necessary for this conversion into the toxic metabolite can be increased with an inducer. So we added a toxic compound before we added the paracetamol to increase the sensitivity of the cells, and you can see there you can replicate this behavior. So the cells die at a lower dose than the non-induced cells. So these are like a valuable system for drug-induced liver injury as far as these tests go. Another example, and Maria João just touched on this really extensively, is the alveolar epithelial cells, which are build the air-blood barrier in the most organs. So you have the air, and then you have the bloodstream, and they're separated by this really tight barrier of cells, which are called the alveolar epithelial cells. And in that case, interestingly, we not only have 2 expansion genes, no, we have much more genes that have been integrated in these cells. And this illustrates that we can't really tell which genes and how many are necessary for the immortalization of such a cell line. And that's why we always have this very unbiased approach with this huge library. So these cells, when you plate them on a metric system, so similar to what Maria João did with the A509 cells, then what you see is polarization. And these are immunofluorescence images over a set stack, so multidimensional. And what you can see is that, so if you then stain some tight junction proteins, what you see is that, for example, ZrO1 is only localized to the upper area of the cells, if you look at it from this angle, which means this is the area that would be exposed to the air. And this just shows you these cells polarize, and they polarize in the right way. And then we can go on and do exactly what Maria João did with the her cell line. We can culture them in either liquid-liquid, so submerged conditions, which is the square here, or we can also culture them in air-liquid interface conditions where we have this exposed area. And what you can see is these cells build a barrier in both of these cases. So this is just typical epithelial barrier measurement via the resistance. And what you can see is after 7 to 9 days, the cells start to build a barrier, and in the air liquid, so in the non-submerged conditions, you can see a higher barrier formation. And this is the first step to making these cells valuable for screening purposes, especially if you look at toxicity assays, because then you can do transport studies, but you can also look at the exposure to, for example, nanomaterials and how toxic they are. And so that's the point where we said, okay, we are fine with the cells to give to other people which are much more experienced in screening toxic substances. And these cells have been quite extensively used to model the air-blood barrier and to test toxic substances. And to illustrate this, I'm just going to quickly show you some of the posters and talks at the SDF conference. So the conferences where Maria, João, and I both attended last year. And there was a poster, a good colleague of ours in Berlin, and they look at how our cell line, so this alveolar cell line I just showed to you, stacks up against the typical models that are being used right now as model systems, for example, the A509 cell line that Maria João just talked about, and how they use it for nanoparticle toxicology. And so far, the data looks really promising, so it really looks like it makes a difference what kind of cell source you use. And our cells just seem to be much more sensitive towards these nanoparticles than the A459 cell line. And just to illustrate again, there was a talk of Udo Bock, and just in the group in Saarbrücken. Also, again, really good colleagues, and we established the cell lines together with them. And they also now use the cell line that we established back then to use it in a consortia to validate it as a test system, alternative test system for an animal model, so to use it as an air-blood barrier model. And you can also use them for infection research, so they have been successfully cultured with macrophage THP1 cells, and so they are being used as a model system. So this is basically what we do. So we immortalize cells, we look at the function, and we look at the suitability of these cells for specific assays. But I also told you in the beginning, what we want to have is a reproducible cell source and also reproducible assays. So how does the reproducibility is now? So reproducible cell line generation. What I told you is, so we identify proliferating cells from the fresh primary cells that we infect with this lentiviral library containing 33 genes. And interestingly, what we found is that specific cell lines, they do carry specific immortalizing genes. If you do this process often enough, so you produce a lot of these cell lines and you infect them with this library often enough, what you will find is that specific cell types always carry the same expansion genes as a cocktail. And this is really interesting. For example, for intestinal cells, we found that pretty much 3 cells are more than enough to immortalize them. And every time we do the immortalization, we see the same 3 genes popping up. Similar thing for hepatocytes. So there's a limited set of genes that is necessary and enough to immortalize these cells, and they still keep a good function. And now this is the really interesting part about this technology, is we can take these cocktails once we have identified them and then go back to fresh primary material and always generate in a reproducible manner, again, a cell line from fresh primary material, only using this specific cell type specific expansion cocktail. So we don't need the whole library again, but we only need a specific cocktail, and this leads to the reproducibility. And just to show you an example, so we did this for umbilical vein endothelial cells, and this is the cells that we best characterized in this regard. We looked at the proliferating cells from them, and we saw that they carry 3 genes that lead to successful immortalization. This is ID1, MYC, and ID2. And no matter how often we did this immortalization, we always found these 3 genes popping up. So we took this cocktail and infected, again, fresh cells from multiple donors this time. So 3 different donors, and what you can see in these growth curves is that with this cocktail of expansion genes, you always get cell lines that behave very similar in terms of proliferation. So you always get good proliferating cell lines. And another interesting aspect is, again, so we have this 3-gene cocktail that always immortalizes umbilical vein endothelial cells. And what you can do is now immortalize cells multiple times from the same donor. So we've done this as a replicate. So we have one donor, but then the different immortalizations. And what you see in these flow cytometry plots, where we stain for typical endothelial markers, for example, CD31, acetylated LDL uptake, or the activity of eNOS, you can see with this cocktail over multiple immortalizations, so these are 4 very different immortalizations, but from the same donor, you always see a very, very similar phenotype. And this means you can have very robustly and very reproducible generate cell lines even after, in slightly different conditions, meaning different infections. And this is the reproducibility of these gene cocktails that I wanted to highlight here. Okay, so just to quickly sum up what I told you. So we have a species-independent and tissue-independent approach to generate cell lines. And these cell lines are expandable, but they're also functional. And these are the 2 most important criteria for multiple applications, but a prime example is the in vitro toxicology. Because for this, you need a model system that is widely available, that is really easy to culture, can be produced in high enough biomasses, so it's useful for screening purposes, and it needs to be really reproducible. And that's what we hope we can achieve it with our technology. And with some cell lines, we've already shown this, and for other cell lines, we are currently in the process of showing this reproducibility and setting them up as alternative models to animal models for in vitro toxicology. And with that, I would like to encourage you to get in touch, share your opinion about cell lines or how useful you think they would be in these 3R approaches and in vitro toxicologies. Ask questions, be curious about them. It's not a typical approach to immortalization or to in vitro toxicology. So we always like to get feedback. So you can reach us easily, you can reach me and just ask questions. Also, you can refer to a few publications, and I'm happy to share them with my PDF file in a couple of days as well. So yeah, thanks very much for listening. Again, thanks very much to ACCT, to the STIF for setting this up and for the chance to present the work. So thanks very much for listening.
Okay, great. Thank you, Tom. We have a question that's come in already. Just a reminder to everyone, if you have a question for Tom or for Maria João, just type it into the question box and I can read it out for you. The first question is, are these clonal cell lines or mixed populations? If clonal, at what point in the process are they cloned?
Yes, so we do both. It really depends on the cell source. So if we have a really pure cell source, then we don't really need to go for clonal cell lines, because what we observed is, and this is empirical evidence over many of these immortalizations, is that at some point there is some cell line in this polyclonal mixture that will dominate in the end after a couple of passages. So for really pure populations, we tend to go polyclonal because it's easier, and sometimes it's not really relevant for the assay. But for example, there are cell lines where we really early on in the process start to go monoclonal. And really early on, I'm talking about 3 to 4 weeks after adding the lentiviral cocktail to them, we try to get them monoclonal, which is sometimes a real challenge because some of these cell lines just don't like to grow as a monoclonal lonely cell line in the dish. So it's really a challenge, but we do both approaches, and this always depends on the desired outcome. But yeah, we do both approaches.
Okay, thank you. The next question is, immortalized human cell lines often demonstrate an embryonic gene expression phenotype. Have you compared the expression patterns in your cells with those of adult primary cells?
Yeah, really good question. We did a bit of characterization in the Nature Communications article there, looking at gene expression patterns. So in the supplements, you will find a microarray analysis where we compared some of our immortalized cells with the primary source material. So this might be really interesting, and yeah, if you'd like to know more details, you can just write me an email and I can look in the gene expression patterns. So we did the comparison for the specific cells we looked at. In this case, it was also the endothelial cells. We found they really preserved the adult phenotype, so they didn't really go back to an embryonic phenotype. But this was in this case also due to the fact that the immortalizing genes that were integrated in these specific cell line don't really have a stem cell kind of background. So as I mentioned earlier, we have some genes that are also known as the Yamanaka factors, so they're reprogrammed cells, for example, KLF4. So if you would have a cell line integrating a lot of factors that confer stemness, you would assume, okay, they would go to the back to the embryonic phenotype. But in general, what we see is our cells, they really preserve the adult phenotype. And we always have the advantage that we get a good amount of cell lines. So usually we get 10 to 50 cell lines out of our immortalization, and we can then look at the phenotype. So even though we might have some embryonic-like cells in there, we wouldn't really consider them because we just would look at the phenotype and the function, and then go back and only select these cells, not the embryonic-like cells that we get.
Okay. How about the possibility of excessive gene expression during the assay, considering the unlimited proliferation of the relevant cell line?
Okay, so I assume this is about the expression of cell type specific genes, or is this question focused on the expression of the expansion genes itself? So if this could be clarified, then I think I would be able to answer the question more clearly.
Well, let me try unmuting the questioner, and let's see.
Otherwise,
we should. I've unmuted you if you want to clarify your question.
Okay, so for example, like I'm working with the liver cells. So like you said, like usually cell line they're expressing to certain kind of gene expression, like albumin, for example. So since like for this relevant cell line, they also have a high proliferation, just like the cancer derived cell line. So do you think that, for example, like they will express like excessive albumin expression, something like that?
Okay.
Like during the assay, yeah, because when you do the like the hepatotoxicity, like the toxicity assay, so there's a possibility probably the albumin expression is really high. So when you do some like treatment, maybe like like the expression is not gonna be affected that much.
Yeah, really interesting question. Actually really relevant because I worked with these cells during my PhD thesis, so I was wondering myself. So two things. So first of all, the cells that we generate, in the majority, they are not per se transformed as in cancer cell lines. So we did some assays where we implanted these cells subcutaneously into mice to see whether they would form tumors. And we tested 7 cell lines, and we only had 1 cell line that formed tumors. So these cell lines, and that's why we call this kind of also soft immortalization. So they're not per se transformed. And then for most of the assays, what we try to do is we don't really want to have them in the proliferative state, but what we often do is provide a brief stimulus, for example, 3D culture, or putting them on, as for the alveolar cells, putting them on the transfer membranes to kind of provide the stimulus and get them away from their proliferation a bit. But you're right, because especially for the liver cells, albumin is sometimes also called a fetal marker rather than a really adult marker. So, but yeah, so the expression was in line with fresh primary cells. So I showed this on one slide. So if you look at the expression, it's a bit lower than primary cells, but you can reach primary cells level. But yeah, so this is a problem. I mean, hepatocytes are incredibly difficult to immortalize really successfully. And we were quite lucky with the cell line. And also we found that providing a stimulus, in this case, a 3D, really helps with the gene expression, controlling the gene expression pattern.
Okay, that's interesting. Thank you.
Thank you.
So we have 2 more quick questions, hopefully. First, what are the functions of the various critical genes responsible for immortalization?
Yeah, really good point. So it's a mixture. So as I mentioned, the 33 genes, they are either of human origin, of murine origin, or some viral genes as well. For example, the human papillomavirus E6, E7 genes. And we have some anti-apoptotic genes in there, but we also have genes that control the cell cycle, or they have to do with DNA damage, or they have been known, for example, as I mentioned, human papillomavirus E6 and E7 to mess with the proliferation of epithelial cells. So yeah, we have various functions that either control cell cycle, apoptosis, senescence. In the publication, again, in the publication, in the Nature Communications, you can find a list of genes. So all 33 genes are listed there in case you're interested, or just write me an email and I can elaborate on some of the functions of the genes. But the list is publicly available, so it's no black box. And yeah, they have different functions on cellular proliferation, but in the end, it all comes down to cell proliferation. They are related to that in some or the other form as well.
Okay, and the last question, do the clonal cell lines give rise to mixed populations after several weeks of culture?
Sometimes they do. So we've seen this phenomenon where we have bona fide clonal cell lines. So always with cloning these cells, it's really difficult whether you have a pure clonal cell line. Mostly because we now have a lot of experience with doing that. So there's not really a specific trick that we do. It's just a lot of experience that we, and especially one colleague of mine now has. But what we sometimes see is if we want to immortalize epithelial cells after some weeks, even though they are supposedly monoclonal, you have some sort of fibroblastoid cells popping up. And what we don't know yet is whether this is an original contamination that was there to begin with, so the cells were that monoclonal, or what we also now consider a possibility is that some cells undergo some sort of epithelial to mesenchymal transition later on in the process, and these cells become fibroblastoid. So this happens in rare cases, but that's why we always include the stability control. So we always have the cells running for at least 2 to 3 months before we say anything about their pure population, just to exclude we have any contamination or we have any instability in the system. And again, we have the advantage that we have 10 to 20, 50 cell lines, and we can look at a lot of them and see which one is best and which one doesn't show any instability. But this can happen, yeah.
Okay, great. I just wanted to thank you for answering all those questions. Some really good discussion there. And I'm going to go ahead and close the webinar. I just wanted to thank both of our presenters, Tom and Maria João, for their time. I really enjoyed learning about your work, and I know our attendees did as well. If you have any questions, feel free to get in touch with me to follow up from the webinar, and we hope to see you again next month. Thank you very much.
Thank you very much.
Bye-bye.